So again, for those of you who have been here before, Dr. Foster is not going to be a stranger to you. But for the new families, Dr. Foster is located in Hamilton at McMaster University, and she's the lead of our immune platform. So we talked about in the morning how we are not restricting our questions to the brain, because we know the brain talks to the rest of the body, and the rest of the body has, the way the rest of the body functions has impact on our behavior and our thinking skills. And so immune system is one place where we have made some investment, and we're going to talk a little bit later about all the things we need you to do. But for now, I want you to get excited about the science, and welcome Jane to the podium.
So it's always a challenge being the person who's between the audience and lunch. So I strategically prepared a shorter talk to give both an opportunity to get us back on schedule or closer to the scheduled time, but also because historically there's always a lot of questions. And so I'm going to give you a little bit of information about how your immune system talks to your brain in general, and then I'm going to tell you some specifics about what we're actually doing in the immune platform, from what are the things we're going to measure there, and what blood do we have to actually take in order to ask these questions. So a little bit of practical information about what exactly we're doing in the platform. And so I like to start and think about this. This applies to everybody in the room, right? When we think about the immune brain system, it's part of healthy everyday homeostasis. It's how we develop early in life. The immune system is integrated into all systems of our body, and that's why it's such a great biological marker of health. And so what I'm talking about here is not when you get an infection. This is about everyday immune cells, immune molecules in your body and in your brain that contribute both to behavior and to brain function. So this is a continual communication, and it's bidirectional, right? Everything about our behavior is driven by our brain. It's the master communicator that tells your arm to move, right? And the way that your brain connects with your immune system is through some of these nerves in your autonomic nervous system, but also through chemical release, through your pituitary release of hormones and other things. So your brain can communicate with your body, and your body can communicate with your brain through a whole host of ways. And here I list neural, humoral, which is in your blood, or cellular. And I'm going to talk a little bit about a few of these things. Very important to brain development. In the last 2 decades, we've learned so much about how your immune system is important to healthy brain development. And I'm going to give you one example about the immune cell in your brain that some of you might have heard about before and some of you might not have. It influences behavior. So one of the examples I can tell you that we've done in both animal studies and in clinical studies is it seems that general inflammation, so that's the amount of immune activation that's going on in your body, as that increases, you get increased anxiety. There's a very good relationship between increased activation of your immune system and increased anxiety. And we can take that from infection right up to the other direction. When we reduce inflammation, we get reduced anxiety. So it's a continuum of normal, healthy body. And anxiety is just one good example. Your immune system has a whole host of molecules and signaling pathways. Those molecules and signaling pathways are present in your brain. They do some of the same things in your brain, and they do some unique things in your brain. And so this concept of immune privilege that is actually about 150 years old, when somebody discovered the blood-brain barrier, is actually a little bit of a myth, because your brain is completely open to communication from your immune system. So I'm going to give a couple of examples, and I'm working on an infographic. I didn't get it done for today, but I'm willing to take any feedback as I move there. This is my drawing, so it's a little bit brutal. But I did draw this myself on my computer. And these microbes over here are actually accurate to the EM level, right? These are real microbes that we have up there. So I'm going to talk about 3 different areas of research that would be considered under this immune brain research. I'm just going to give you a couple of examples. So one is up in the upper corner up here, is this microglial cell, which I think needs some improvement. My neuron's pretty good. And this glial neuronal interaction is an important part. It's not something we're studying in our immune platform, but it's something we study in our mouse studies. It's a very important part, and sometimes people think that the neurons are the only player here. So Steve told you about some of those targets that wire synapses. Those microglia are the ones that actually receive the signals. They're called eat-me signals that actually help refine those connections in development. So the microglia is your immune cell in your brain, and it's very important to how we wire our brain. I think I probably have that on a slide, but I already said it. Your immune system and your gut are interlinked. I'm going to talk a little bit about that, and sometimes, some of you have heard me talk about that before. If you're interested in microbiome-related stuff, you could go to my website. There's a lot of information there. And then I'm going to talk about these particular immune cells, because that's one of the things we can measure. We can measure who's there, what they're doing, and one of the things that immune cells do is they produce these inflammatory mediators into the bloodstream. And so these are different things that we can actually measure as well. So microglia are the resident immune cell in your brain. They're not generated in the same way as your peripheral immune cells. They're generated from the yolk sac during development, and they have a self-renewal during life. So they're very dynamic, they move around, they're continually active. When people do in rodent studies where they image these through using different microscopic techniques, they change their processes continuously over time. So they're very dynamic. They're very important, as I said, to building those synaptic connections, who stays and who goes. One of the things that's fascinating about microglia is there's completely different profile of microglia and how they respond to challenges in male mice versus female mice. One of the areas here in Toronto that has done some great work here is showing in the response to pain. Right? So Mike Salter's group here at Toronto has shown very clearly these microglia are very important to how sex differences in response to pain happen. So when we start thinking about different neurodevelopmental disorders that might have a bias with respect to gender, the immune system might help us tease out some of the different biologies that might be going on there. Infections and other insults activate microglia. So these are still an immune cell. They're every day doing something not very infectious related. They do all these other important things to your nervous system, but in the end, when you do get an infection, they also turn into what looks like a macrophage in your brain, and they can both have a beneficial effect there, but in disorders such as Alzheimer's disease and some neurological disorders, an overactive immune system at the level of your microglia can actually be damaging. So your immune system is there to protect you, but if you have a bit too much, then it can be a problem. Shifts in the relationship and the conversation between your microglia and your neurons actually changes behavior and might influence the trajectory of brain development. So when we're thinking about the brain itself, we need to remember that the microglia is an important part of the story. I'm just going to do one slide on the microbiome. So all the surfaces of your bodies are covered with microbes that could include bacteria, viruses, fungi, protozoa, parasites. But bacteria are the best studied microbes. We studied those in both mouse models and in clinical populations in my group. There are trillions of viable bacteria in your gut. There's very few organisms from the worm through to humans that don't have a microbiome or don't have a bacteria that's helping them through their day. These are essential to the development and function of the immune system. So we get our microbiome as we are born. This relationship starts at birth, either vaginally by gobbling up those bacteria, or through the womb or a breast, depending on mode of delivery. And it's critical to the function of your immune system. So some of those allergy problems and asthma problems that occur when there's problems at birth are linked to this disconnect between your microbes and your immune system. So without microbes, you don't get a normal immune system. But also, we've got some recent data in some of our mouse studies that show that if you take away a component of the immune cell, which we can do in mice, genetically modify mice, you actually get a completely different profile and functional microbiome. So these are, again, bidirectional conversations that your body's having, and those have a bottom-up effect on your brain. So there was a question in the list, it's the only question I'm able to answer, about are there effective treatments for autism involving the gut? And the answer is Yes, potentially. So there's a lot of evidence that the microbiome composition and diversity as well as function has changed in individuals with autism, and that might, I think there's now at least one OCD study as well. But these are just who's there. This is not really, telling you that's the problem. It's just an observation. Most of these are observation. There is a lot of GI disturbances in individuals with autism. I put this up here because I think these are two different things. Again, like when I tell you that the immune system is part of normal, healthy development, the microbes are part of normal, healthy GI motility and all those other things. So you don't actually need a GI disturbance in order for your microbiome to maybe be influencing your behavior or your brain function. If you do have a GI disturbance, you're going to have a disruption in your microbiome, and it can be part of the story of how that's working, and how it might affect your immune system. So those are important considerations, but they're sort of two separate components of it. There is a lot of ongoing research on microbiota-targeted strategies. Diet interventions, diet is a big driving force of your microbiome and of your immune system. Probiotics, and then fecal microbiota transplant people often ask me about. The best evidence for this is actually, as it sounds, taking somebody else's poop and giving it to a person. It can be following, depleting their own system with antibiotics or without. There's both strategies that people employ. The best evidence for that is in C. difficile. That's where it sort of got its legs, so that for C. difficile infections, if people are treated with or don't respond to the normal treatments and they actually get a healthy fecal microbiota transplantation, it can take away that infection. So that's the best medical evidence of this. In all sorts of areas, people are experimenting with this. The best place to find that evidence currently is in clinicaltrials.gov. That shows you who's doing what, but not all of those studies are producing answers yet, and it's not a please don't do this at home. Right? This is, I can see Evdokia dying when I'm trying, she's dying here. So it's not something that you can employ without medical help and medical recommendations, right? So you can consider probiotics on your own if you'd like to look at the evidence. And I put this website up here because it lists all of the probiotics in Canada that are available, what their contents are, and what's the clinical evidence of their use. So what we say about these interventions is you should use the product that has been clinically demonstrated for the indication that you're trying to treat. So if it's a GI disturbance, there's a list there of the ones that work in GI. There's a pediatric set that has the ones that work in kids as well. So it's a very good reference, and it comes from the International Society for Prebiotics and Probiotics, which is an academic group of academics who really promote education related to prebiotics, fermented food, and probiotics. And so they're exceptionally good. On their website, it's a whole host of infographics. I have one on microbes and mood that I can share with people. But that's really good information. Okay, so back to the immune system. So the peripheral immune system can communicate with the brain through all sorts of mechanisms. So it can actually produce a whole host of immune mediators. It can actually talk to your sensory nerves and send a signal up to the brain. They can release things into the blood that can target the brain, and the cells themselves, these immune cells, move around, and they can actually both traffic into the brain under certain conditions or just come into the close proximity of the brain and release molecules that have an impact on the other side of the brain. So there's a whole host of ways. I give a whole course on that. You can come to McMaster and take my class, on how your immune system communicates with your brain. Circulating immune cells influence normal brain development. That's why when we activate the immune system at different stages in development, there's some risk associated with that because it's part of normal brain development. Individual differences in the immune system are just as great as the individual differences we see across the room. So our biology, as I'm sure Evdokia spoke to at the beginning, underlies some of our individual differences, and one of the places that we might be able to identify biotypes is in our immune system, and that's what the immune platform is trying to do. And we can measure certain features of an individual's peripheral immune system from a blood sample. So that's a sample of convenience. for measuring these things. You can measure some aspects of the immune system using other samples like saliva and other things, but for our purposes, what I'm going to show you is what we're going to measure, and it really gives us a better biological readout than from the functional aspect of what your immune system's doing. So instead of giving you all the things that are happening in this field, because it's a fast-moving field, I've just highlighted a few news articles that are just in the last month, right? These are all in October. So, inflammation and autism, an important piece in the puzzle. There's research going on all over the place. In my lab, we train high school kids in science, and what we do is we have them tell the research story from the news. So they, find the news, and then we help them understand the evidence behind it. So if you're interested in this area, Twitter and the news feeds, as long as it's, good news, is an actually good place to sort of get a little bit of a primer, as long as you remember the source that it's coming from, right? It's good if the article has a reference to a paper or a researcher or cites the people that are doing the work, but it's still a good avenue. Researchers received $3.2 million NIH grant. As Steve said, we'd have a lot more money if we were on the US side. We can apply for NIH grants, but I don't think it's as easy to get them over here, to pursue a deeper understanding of Down syndrome. And this is actually targeting the immune system in individuals with Down syndrome. So this is a first from my perspective of a large grant funding taking a look at the periphery and the body and how it might help out, particularly to the concept of precision medicine that Steve referred to as well. Brain's immune cells may protect against OCD, anxiety. As I told you earlier, this link between gut immune anxiety is robust across animals and people. And another the microglia itself, is what both of these news headlines are referring to, that changes in the microglia can actually benefit anxiety. So it doesn't always have to go through that nervous system neuron. These other supporting cells that are highly dynamic and respond to all the environmental changes can be a place where, for example, exercise has a benefit, right? Can be through activation of some of these immune signaling cascades. So there's several benefits of looking at what we would call the immune phenotype in brain health and in neurodevelopmental disorders. First is these biomarkers. So understanding biological difference to help subgroup individuals based on their shared biology. I'm sure Evdokia covered that. Provide new biological targets for the development of new drugs or therapies, right? So even if we can, help identify who's going to benefit most from an intervention by their biology. The immune system is a great place to start. And how does it change? So if we're seeing some people benefiting and some people not benefiting, is it because those people are benefiting have a positive impact on their immune health? Expanding these diet and other intervention strategies that I mentioned for neurodevelopmental disorders. And this precision medicine approaches, which just needs an education of what should we be using in our biomarker kit in order to advance these things. There's a lot of interest both in the diet intervention, nutritional psychiatry, but also in classic pharmacological approaches with respect to the immune system. And so the work we're doing in POND actually looks at some of these cells, and I'll just tell you a little bit about the ones that we're doing, and then I'm going to tell you what blood we take. So we can look at the peripheral immune cells, and we do this using flow cytometry analysis, which means we actually take the blood and put a whole host of antibodies in there that stick to the surface markers on your blood. So your blood cells have particular proteins on the outside of the cells that are actually specific to a type of blood cell. That is also a protein that can tell us how mature are these cells and what are they doing. So for example, macrophages that can move out once there's a signal that there's a problem, they move out of the blood and into the tissue. But the proteins they present when they're moving or exiting look different. And what's really interesting is those macrophages that go to the brain look slightly different than those that go to the gut, for example. So we can actually get a lot of information about what the function of your immune system is doing by looking at something like the monocyte. And these are, I'm going to show you the tubes later, so I'll skip over the sample prep. Cytokines. So cytokines are immune molecules. They can both be more activating your immune system or dampening your immune system. And we use protein assays, so we can take a plate that's coated with these antibodies, we can put a sample in it, and we can measure how much of these proteins are these. So these are the signaling molecules of those immune cells. Other tissues can also produce inflammatory mediators such as cytokines. So, the source of it is not always the same. So it's nice to know what are the blood cells there and what are the products that they're making in the same sample. So that's one of the strengths of our platform, is that we actually look at the cells as well as the products of the cells in the same sample. And these are, you have plasma samples for those. And then in that barrier between your diet and your body and your thing is that mucus layer that lines your intestinal tract and all those microbes. And when that is not as strong as it is, you sometimes have transport or increased inflammation. So you can actually get bacterial products in the blood, and we can measure these in the lab. And so we can also measure some of the host enzymes or proteins that are involved in keeping that barrier intact. And so these are sort of gives us a little window into whether if some of the increased inflammation we might be able to observe is actually acting at the level of the gut, or is it coming from something else. So this is what, if you enroll as a child or a young adult in the Pond Immune, which now endocrine platform, I'm not sure if Evdokia spoke about that, and I'm not going to speak about it, but the endocrine component is your hormones, and how do those hormones influence behavior in brain, and that's just launching. We've merged the two research questions together. So we take a couple of different tubes of blood. So we take a small purple top tube, which has a bit of EDTA in it, and that we send to the classic clinical lab. So that would be what's referred to as a clinical white blood cell differential. So that actually just is a quick clinical test to say what cells are there. And that's sort of a control if you're doing any work with peripheral blood and trying to imply how it's impacting your brain. People always criticize you because if you have a different profile of immune cells in your blood, you could have a different biology happening. So that's an important sort of control for blood work when we're comparing across people. And then we take one vial or two small vials or one large vial of a green top tube, and this we get three things from. So this is really, we take a process of processing that blood through a gradient, and we end up getting plasma, which is what I can measure cytokines and other things like that in, bacterial products. We get these peripheral mononuclear cells. So this is a subset of your blood cells that includes those adaptive immune and innate immune cells that are actually doing the work in response to an infection, in response to other things, and are in response to this normal, healthy balance. And then we get the other immune cells, which are called the granulocytes, which are those more reactive cells that might respond to allergy and other things, although neutrophils are part of your innate immune system as well. So we get all three of those from that one tube, and then we took one tube of serum initially, so this is just an empty tube. We let it clot, and we take that rich part without the cells. So it's a little bit different plasma and serum, and different things are stable in different ways. So we get both of those because there's different assays you can do on those two different things. And most of the work in the serum is likely to land in that endocrine hormone part of the question. So you come in and we take all of this blood, and then we request you come back 2 months later, right? Or 3 months later. And this is important for two reasons. One is it takes a lot of sample to do these tests we do. So, we freeze everything, and we've now established that we can freeze all these things and thaw them even up to 2 years later, and everything's viable for our assays. So we're able to do this flow cytometry 2 years later from the bank samples. So if something new comes up, we can go back to our bank and have another look. But the 3 months later is really important because, as I've been telling you, these are dynamic molecules and dynamic cells. So if I take your blood today, it's a snapshot into what you ate this morning, your general health, and all these other things. We don't want biomarkers of breakfast, right? So we want biomarkers that have more stability. So we need reliability and we need validity. So we need to test a second time to make sure when we discover the right biomarker that it's a stable marker that's always going to represent that link to behavior and brain function, regardless of what you eat or whether you exercise that morning or all the other things that can influence your immune system. So it's a very important step in our process to have both of those samples in order to get the best information. So the samples are banked in my lab at McMaster. Don Bowdish is the other person, immunologist at McMaster that works on this platform as well as all of our coordinators and all our leads across all of our sites. We have over 300 samples banked to date of all those different profiles that I told you. We've actually analyzed 270 of the cells using a monocyte panel for numbers, maturation, and function, and that's being analyzed. And we've also analyzed about 200 of some of the samples for some of these inflammatory mediators. These are across all the disorders and the healthy controls. So the idea is very agnostic to the biology, both in healthy individuals and in individuals with neurodevelopmental disorders. There could be a link between immune phenotype and behaviors, is the idea. So I'll stop there and I'll take any questions.
Request before we get going. We have lots of people who are joining us from other parts of the province. And if you don't use your mic when you ask a question, they can't hear you and they can't figure out what the answer matches to. So let's stick with the plan, and if you get any questions outside of the mic, please repeat the question.
Okay. So we have the red one that's not blinking is the person who has the mic. Yeah.
I was just wondering what you know about the consumption of sugar and its impact on like the kid's immune system with ADHD and ASD.
So sugar does a lot of not good things for your gut bacteria to start with. So refined sugars are probably the best way to disrupt your bottom-up component of things. Direct effects on a whole host of endocrine and other pathways probably have direct effects on your immune system. I don't know of a direct change in profile from that, but I'm not sure that people have looked. I could look it up, though. I don't think clinically there's not a...
I mean, glucose changes brain metabolism, right? And some of it is related, but
yeah, so there would be transient effects from any diet's perspective, but I don't know of a stable change. But the gut for sure has a huge impact on your microbiome.
Thank you for speaking relatively slowly. I don't know any scientists myself, but you all speak very quickly.
That's a good prize to win because I normally speak at the speed of light. Yeah, I don't know who has the non-flat. Oh, there we go.
So thank you for that slide you were showing on researchers received $3.2 million, the NIH grant to pursue a deeper understanding of Down syndrome. I'm curious, in Canada, what are we doing in the area of Down syndrome? And more specifically, where does the genetic condition Down syndrome intersect with the neurodevelopmental disorder of autism?
Wow. So I think Evdokia is probably better to take that than I am. We do have at least one individual in our cohort so far.
I should take this one because so theoretically Down syndrome is part of the disorders that we are including in PON. We haven't been very successful in getting the families to come in, so we have very small numbers. And it's very hard for us to do meaningful research when the numbers are very small. So if you're representing that community in some way or form, we would love those families to come in. There are all kinds of reasons why we want, first of all, as we said before, lots of genetic conditions have developmental phenotypes that may not meet a particular diagnostic category, but we are still interested because we're trying to understand how biology links to behavior, right, irrespectively of any labels. But to your specific question, rates of autism in Down syndrome are 10 times that of the general population. So we know that they are much more likely to get autism, and they almost all have intellectual disability. Not all, but there is a shift on the average on cognition, so that the majority of people with Down syndrome have intellectual disability. So there's all kinds of reasons why we want Down syndrome to be part of POND, but we are still very small numbers.
I think we're at the front over here.
Oh, I'm ready. I was just wondering, I mean, I've noticed in my son, he's got a very high pain tolerance, and his immune system too was always compromised. He had RSV at a couple days old, was not hospitalized, but later I found out what that kind of meant. And then he had strep as a carrier. He never got symptoms. So he never got, I don't think he had to learn to blow his nose till he was 14 because he just never reacted to anything. Is that
So
tied to-
the anecdotal stories of things that look a bit different are prevalent. I've heard a lot of them over the years when I give talks, particularly in public crowds. And the thing is, as I told you, your immune system does a lot of different things. So you can have, like your T cells are important in wiring parts of your brain. Your macrophages are communicating to all your tissues about your state. So there's several levels where you can have a compromise, and that might not land in an infection domain. So the infection is sort of the iceberg that you see. There's a whole host of other things happening in there. So sometimes these other examples, I didn't go to medical school, so I can't comment on the specifics of the medical condition, but these other examples I think actually give more credence to this concept that maybe we can use the immune system to sort out the biotypes, because that will be a very different biotype than the individual who hasn't had that life history to date. And potentially we can measure that if we find the right biomarker.
And is it related to, well, I've heard things about related to allergies, which we have tons of in our family, and to chronic fatigue in the parent and the mother.
So there's we have to remember, Steve gave you a really good outline of some of the advances in the gene side of things, and with all of your immune system, your microbiome, all of things, they're imprinted we're hardwired by our genetics. But all of these other factors influence how it ends up in the end. So at one point we thought some of these differences we see are written in stone. We now know that all of these things have a continuum. So your genes wires you, both your brain, but also your immune system and your gut. But the other things you do, mode of delivery, diet, early exposure to particular pharmaceuticals, non-antibiotic or antibiotic, those all influence your immune system during a point where it's developing. Right? So we're not born with a functional immune system. It actually happens in that early part of life. So if you disrupt things during that early window, you have a different trajectory. Sometimes that's a resilient trajectory. Sometimes that's a risk trajectory. And those are the balance of things. Yep. Last question. I'm not sure who has the. Oh, I think it's in the middle of the room.
Hi. I'm with respect to work that. Like Paul Patterson's work about how maternal infection can lead to autistic behaviors in animal models. Has anybody been tracking that in humans, like looking at history of maternal infection?
So environmental
in moms
causes,
and whether this actually predicts?
So some of the work that we've done in my own lab and in clinical populations, people have done it as well. So it's not just infection that's a risk. There's a whole host of there's drug effects in utero that can have an impact on neurodevelopment. So from my perspective, again, where I talk about GI disturbances and microbes, this is two different categories. What are the causes and the trajectory that land you in a place where you might have a dysfunctional immune system, which might contribute to symptoms or symptomology or causation in neurodevelopmental disorders? There's no debate that there are environmental factors that add to your genetic risk and potentially independent of that. I think Steve would probably tell you there's some genetic risk in there that the environment is adding to, and I would probably agree with that these are the immune dysfunction is a modulator that exposes some of those risks that are written in our genetics to start with.
Okay, so you think that
there's a whole host
you already
of
have
work.
to have a risk factor genetically before, say, a maternal infection could turn on.
I think that we have differences, individual differences in our genetics that underlie how we respond to the environment differently.
Okay.
Right? So whether you call them risk or you just call them your individual phenotype, right? Differences that we have, right?
So the only piece I can add from the human data is that we have some evidence that in periods where we have pandemics, so like a flu pandemic, or we get an increase across all neurodevelopmental disorders for that year of birth. But it's a small effect. So again, it goes back to this gene by environment. It's unlikely that the infection on its own is adequate to get you over the threshold, but more kids cross the threshold on years where we have a pandemic than in years that we don't, which is kind of a little bit of indirect data, but it's still a small effect. Yeah. Okay, so before lunch, and again, Jane will be around, so lots of time for questions. Bug all the speakers. That's why they're here. But I want to introduce you to young collaborators. So we have Joshua and Lucas. Where are you guys? They're going to come down and introduce themselves, and then they have a station outside where they can talk to you about what it's like for them to have been part of research and part of Pond, and where they see the value.
Thank you. Good afternoon. My name is Joshua Evangelista, and I am 17 years old and a participant of the PONDS research study. Here with me is my friend Lucas, who is 15 years old and also participant of the research study. Lucas, do you want to say hi? Oh, he's a little shy. We are here today to talk about our experiences with PONDS and our brain scans. We hope you will come and speak with us during your lunch break. We can try to answer any questions you may have about participating in the brain scan research or even about our diagnosis. Thank you for listening, and we hope to meet you at lunch, and we hope you enjoy the rest of the day.